So I will talk you about hERG channels. hERG channels belong to the same family as hERG, the KV10.1, first explained by Louise. And it is, relevant and famous for different reasons, because it regulates the cardiac potential. hERG sustain the IKR, so the rapid repolarizing current. And then for this, it also regulates, its activity regulates the QT interval. And for this reason, it is considered an anti-target because several drugs which can inhibit hERG also lengthen the QT interval and then can cause an arrhythmia, a ventricular arrhythmia, which is named torsades-de-Pointes. So many hERG blockers are arrhythmogenic, are cardiotoxic, and all the drugs must be tested for their capability of binding and inhibiting hERG. But nowadays something is changing, but nevertheless, they must be tested for their activity. But since a lot of years, so many years ago, I don't want to look at the date, and I discovered a very unusual current that I named a novel inward rectifier and potassium current. It was not an inward rectifier, but I discovered afterwards, in neuroblastoma cells. And this current has a very clear characteristics. The activation curves were dependent on the cell cycle phases. So it was unusual for us. And we started going on and studying this type of potassium current. And then afterwards, some months later, a group in the US discovered that it was hERG, and hERG sustained the IKR in the heart. And since the 95, we were able to detect hERG expression in different types of human cancer, like the KV10.1. And we can discuss why the two channels which belong to the same family are so overexpressed and misexpressed in different types of cancer. And then here is reported the main types of human cancer where hERG is expressed, the role, and we detected the role of hERG in vitro using cell cultures, cell lines, or primary cells for human cancer, and using, classical approach, inhibiting the current or silencing the hERG protein. Or blocking with other tools, but nevertheless. And then we exploited some antibodies which we developed in the past during these years to try to understand the clinical potential of hERG expression in these different types of cancer. I make an example. For example, in colorectal cancer, the function is to regulate the migration, the invasiveness, the secretion of an angiogenic factor. And when we look at a clinical sample, we detect that hERG expression is a negative prognostic factor in early stages colorectal cancer, stage 1 and 2. And whereas it is a positive factor, prognostic factor, for metastatic cancer, metastatic colorectal cancer, which are treated with anti-VEGF antibodies, bevacizumab. So the clinical potential is relevant, but it's different depending on the type of cancer. Another example in pancreas. In pancreatic cancer, which derive from the ducts, so pancreatic-derived adenocarcinoma, hERG is a negative prognostic factor. Here's again in early stages cancers. whereas it is a positive prognostic factor in those pancreatic cancer which arise from endocrine pancreas, so the neuroendocrine pancreatic cancer. So this is the scheme. But in the meanwhile, while studying the role in the cells, we also got results in preclinical mouse models, and we were able to show that the inhibition of hERG, here again, with drugs, specific drugs which block the current, or silencing hERG in the cells with xenografted, inhibit the growth of the tumor and metastatic spread. These are several papers on this. This is just an example in gastric cancer. And what is relevant from my point of view, I'm an oncologist, a hemato-oncologist, is that if you treat the mice, and we know that treating mice is different from treating patients, of course, but and you combine hERG inhibitors with classical chemotherapeutic drugs, or in this case, VEGF antibodies, anti-VEGF antibodies, the hERG inhibition can potentiate, can collaborate with classical antineoplastic drugs. So we had proposed in the past to target hERG in primary human cancer, to treat the patients with hERG blockers. But that is not possible because many of hERG blockers are cardiotoxic. And so how to be able to target hERG without cardiotoxicity in cancer patients? We envisage two strategies. One is to use the so-called non-torsadogenic hERG blockers. So those hERG blockers that, for different reasons, do not cause the ventricular arrhythmia, the torsades one. Or to look for differences in the tumor hERG compared to the cardiac hERG and target the tumor hERG and not the cardiac one. Use of non-torsadogenic hERG blockers. One of them is clarithromycin, is macrolide antibiotics. We know that it blocks hERG, but the effect on the QT is, on lengthening the QT, is not so strong to cause ventricular arrhythmia. We show that macrolide antibiotics like clarithromycin or also erythromycin have an antileukemic effect, because they trigger the autophagic flux, and they block this autophagic flux at a certain point which goes towards apoptosis. And this occurs because these macrolide antibiotics could cause inhibition of hERG. And this was proven also in preclinical models. Mice injected with leukemic cells, and you can see here that you have a good effect of clarithromycin, and these are the survival curves. You can combine clarithromycin with chemotherapeutic drugs, cytarabine in this case, and you obtain a good improvement in mice survival. And the same if you combine erythromycin with dexamethasone in steroid-sensitive leukemic cells. But what I think was most relevant for this point is a recent paper from a US group, and Gail Robinson, one of the main scientists studying hERG in cardiac cells. So they show that hERG is expressed in glioblastoma cancer. So glioblastoma is a brain tumor, very aggressive brain tumors. hERG is expressed. You can distinguish tumors with low and with high expression. But what is important is that they made a retrospective study showing that those patients who were treated for different comorbidities, depression, seizure, with non-torsadogenic hERG blockers, they have a better survival. And this was related to the high expression. So as more the expression of hERG was in the tumor samples, as better was the survival. We know that this type of tumor has not good survival anyway. So targeting the molecular difference between tumor hERG and cardiac hERG. Here again, two strategies, sorry. One was the preferential expression of the hERG1B isoform in leukemic cells. hERG1B is one isoform of hERG, which has a shorter N-terminus and a quicker inactivation and quicker different biophysics. So we contributed to show over the years that leukemic cells do overexpress this type of isoform, pediatric lymphoblastic leukemia, and another group in Germany showed also in acute myeloid leukemias. And so we developed, along with SME in Germany, a pyrimidine-indole compound, CD6130, which preferentially inhibits the hERG1B, has a good, strong anti-leukemic effect in vivo, in preclinical models, and had not effect on the cardiac action potential in guinea pigs, and did not lengthen the QT in the same model when injected intravenously. In this table, you have the comparison with sotalol that, of course, lengthen the QT. So we proposed CD6130 as a first-in-class compound that could be used since it blocks hERG1B for treatment of leukemic patients. The second strategies of the second type of strategies, so looking for differences, is the fact that hERG complexes with the beta-1 subunit of integrin receptors in tumor cells. Also in this case, the story is very, very long, and it is relevant for cancer because integrins represents adhesion receptor, and as you mentioned, the tumor microenvironment is so relevant in solid cancer and also in leukemic cancers. So we, at the end, after a long study, we published this year a paper in which we summarized all the aspects of the hERG-integrin interaction. And if I have time, I will show you the results. Do we have time? Yeah? More or less. So the first point, hERG not only co-immunoprecipitates with integrins, but it is strictly linked to the beta-1 subunit. And we obtained good data applying the FRET technique, showing that the two proteins do indeed make FRET. And this occurs mainly where the cells are seeded onto an extracellular matrix, which activates integrin and which activate the beta-1 containing integrins, fibronectin. And the linking between the two proteins occurred on the plasma membrane as well as in the cytoplasm, because maybe the two proteins go together through the Golgi apparatus and the reticulum. So, but this does not occur in the heart. So we tested, in this case, through co-immunoprecipitation of the two proteins, the occurrence of the beta-1 hERG complex in several types of human primary cancer, colorectal, pancreatic, leukemia, acute, chronic, and so on. And everywhere we found the complex, because the integrin is activated in the cancer sample. in the tumors in tissue, but not in the heart. And why? I'm sorry, I make it confused. No, okay. We made an hypothesis that this lack of combination occurred because hERG was linked to a canonical beta subunit in the heart. And so we showed that indeed the KCNE1, which is expressed in the heart, is not expressed in tumor cells. And then we overexpress this subunit, and we found that the formation of the hERG beta 1 complex was inhibited. In other words, this is the scheme. In the heart, we have hERG complexed with KCNE1, whereas in tumors, since tumors lack KCNE1, it is bound to the beta 1 integrin, as if it was its beta subunit, its canonical beta subunit. Okay? So the first point is this. The second is that a particular hERG conformational state, the closed state, favors, I will finish, the complexing, and I will go just to the scheme, okay? So we use different mutants. We show that only when the complex is formed, we obtain a lung metastasis from breast cancer injected orthotopically in mice. Okay, and if we disrupt the hERG beta 1 complex, we hinder, the formation of lung metastasis. So which is the future for us? The future for us is to look for compounds, inhibiting peptides, antibodies, which can block the hERG1 complex to the beta 1 integrin. And we hope this will hinder tumor proliferation and metastasis, as it does in preclinical models, but we can avoid cardiotoxicity. This is our future. We hope we will succeed, and sorry for the length.
Thank you.
These are collaborators.
Nice story. Okay, questions for Professor Arcangeli?
The reason why the non-torsogenic hERG blockers are not cardiotoxic is presumably because they have compensatory actions at other ion channels. So they hit hERG, but they hit other ion channels as well. So why not take the strongest hERG blocker that does not cause cardiac toxicity and take that through some development? Because, of course, starting with new development is going to take a very long time. And they are quite good hERG blockers. And if they don't cause QT prolongation, pick the one that's the strongest hERG blocker.
Yeah, thank you for the question. Yes, it was our first idea. And everyone in any audience told me, no, you're crazy, you cannot follow. But maybe times also change the expectance from the audiences in a scientific meeting. So maybe now is time to follow your suggestion. Thank you.
He usually has good suggestions.
Yeah. Maybe if any one of you can help me in doing that, it could be great.
Any other questions? Can I ask you one thing?
No, you can't.
Okay. Right, on to the next speaker.
No, you can't.
Seriously, this also goes to William a little bit. When you block the hERG in whichever way, and you are suppressing both primary tumor formation and invasion of metastasis. I think William has also seen that. I mean, could it be that it's the suppression of primary tumorigenesis that leads to this apparent suppression of metastasis, or could the two be independent?
If I have time, so I don't want to bore you, but we dissected this Yeah, in this picture and in this model. So we were able to show that for hERG, of course, but it can be applied also to other channels. There are some signals, canonical signal, which are mediated by the ion flux, which regulate through the intracellular signaling pathways, regulate cell proliferation. And other signals which are independent on the flux, which regulate the formation of metastasis.
So it's something to do with the permeation aspect.
Yeah.
Okay.